Welcome everyone to the March ISV webinar series. So before I start and introduce Dr. Forster, we're just going to run through some upcoming events to remind you of the upcoming ISV webinars and events that we have happening in the next few months. So the first seminar series that we have coming in April, the 10th of April, will be the Next Generation Vaccinology webinar series. This is followed by the ISV's virtual mini symposium on RSV vaccines on Thursday, April the 24th. And we also have an ISV April webinar on the 30th of April. That's going to be on the safety and immunogenicity of the live attenuated candidate vaccines against Rift Valley virus in healthy adults. And of course, to remind everybody of our International Society for Vaccines annual congress that this year is going to be held in beautiful South Africa in Stellenbosch in October. Registration and abstract submissions will open on April 1st. Currently, we do have awards for non-LMIC and LMIC ECRs and PhD students, and the deadline's been extended until the 7th of April. And the LMIC awards will be actually in person and will cover the costs of the registration fee, accommodation, up to 4 nights, and economy class travel. For the non-LMIC awards, you can find the information on our website, but they also offer the registration fee and accommodation and economy class travel. We do remind the sponsors to join us at the International ISV Annual Congress to showcase your brand. And of course, to remind everybody to join the ISV because of multiple membership benefits that the Society does offer. And of course, at the end, a big thank you to all of our annual supporters and board members. And of course, finally, for everybody, please follow us on different social media platforms to catch up to all of the latest news in regards to ISV. But today, I do have the pleasure of introducing Dr. Angus Forster from Vaxxas, who is the Chief Technology Officer at Vaxxas and has worked in medical product development and commercialization for the last 25 years. At Vaxxas, Angus has the responsibility for research and development and leads the company's vaccine development programs, including multiple phase 1 clinical studies in Australia. We look forward to hearing about all the ongoing work because I believe the last time we had you on as a panelist was about 3 years ago. So welcome and thank you very much.
Great, thank you, Branka. Just checking you can hear me okay, coming through loud and clear?
Yes, we can.
Great, thank you. Let me just share my screen. It's great to be back and presenting to the ISV community again. Let me just get the screen shared. Okay, and just confirming that you can see the slides in the slideshow mode, Branka, as well.
Yes, we can see the slides, and just a final reminder for all the attendees that you can write the questions in the Q&A for after the presentation.
Great, thank you. So yeah, great to be presenting again. I think as Branka said, I think the last time the Vaxxas sort of spoke to the community was back in 2021, and I actually will recap a little bit on what I presented at that virtual seminar during COVID. As Branka said, I'm the Chief Technology Officer at Vaxxas. I've been with the company since 2012, and we are focused very much on skin delivery. Just a few disclaimers before I begin. I'll skip through these reasonably quickly. At Vaxxas, we are all about the vaccination process. So we believe that both the vaccine and the way the vaccine is administered are necessary attributes to protect against infectious disease, and that the process of vaccination itself can actually play a role in improving the effectiveness and reach of a wide range of different vaccines. And maybe apologies a little bit in advance. Some of the next slides I'm going to go through are probably really stating the obvious to the community and audience on the call tonight, but I guess it's always worthwhile looking back at where vaccination has come from and sort of this history that we've been through and the difference that it's made, because I think certain aspects of that are a little bit under threat at the moment. So again, don't need to tell this audience about the importance of vaccination. It's been better described by people much more qualified than me in terms of the benefit it's made to public and global health. And I guess a painting from the first sort of Western vaccination in 1796, Edward Jenner scarifying the skin surface of the milkmaid's son and then rubbing cowpox vesicle onto the skin to help protect against smallpox infection. Now, since that time, vaccines have been hugely important in protecting the lives of millions and millions of people around the world. This is just a list on the right-hand side of the impact of vaccination in US morbidity. And it's just worth noting, I guess, looking at the measles data there, and obviously recent sort of outbreaks we've seen in Texas amongst children for measles, where there's a vaccine preventable disease. So vaccines obviously make a tremendous difference, but since COVID, I think since I spoke to the audience back in 2021, so vaccination rates, especially for routine vaccination, are declining. And this is just some data from the CDC in terms of vaccination rates amongst older adults in the US, so 65 years and older on the left-hand side, comparing sort of the COVID pandemic years to where we are now. At the same point, compared to about 2021, 2022, about 28 million fewer doses have been sold in the older adult population, showing a general decrease in vaccine use for routine vaccination. And then I did a quick, very simple bar plot of that to make it maybe a bit more apparent on the right-hand side, showing that decreasing trend from 2019, 2020, 21, down to 24, 25, where we are now. And so the last sort of slide on that topic is really, it affects, both, obviously has a huge impact on public health, but also affects the bottom line of companies as well. And this is a sort of statement from CSL for their half-year results, and obviously the number of seasonal flu vaccines sold by Seqirus, especially in the adult population, 18 to 64, was much below their expectations. And that was attributed by the CEO to post-COVID apathy amongst that population, but also reduced vaccine access. And I don't want to point the finger at the needle and syringe. Obviously, we're a company looking at alternative technologies to needle and syringe delivery. And the needle and syringe has been a hugely beneficial device for rolling out vaccines. Almost all vaccines are given through needle and syringe-based vaccination. It's very effective at what it does, delivering vaccines typically into the muscle. But it's really remained largely unchanged in about 170 years. So the top sort of diagram there is from the original patent filing for the needle and syringe. And you can see that it remains remarkably similar to what we have now. And obviously there's been a tremendous amount of improvement and innovation in the area of vaccines, vaccine technologies, different platforms, a broader range of vaccines, moving to different areas, immuno-oncology. But the way in which we deliver those vaccines has, by and large, remained unchanged for a very, very long time. And that has an impact. It's estimated, I think, that up to 25% of adults, so a good number of people on this call, have some sort of fear or anxiety associated with needle-based vaccination, which particularly in routine vaccination can affect and impact the number of people receiving those vaccines. So at Vaxxas, we think there's a better way to administer a vaccine using a simple to apply skin patch. I've got one of those in front of me. Hopefully you can see it on the camera, maybe from the screen. I can't see my camera image. But it's a simple to use device. The patch itself is contained in a single-use auto-disabling disposable applicator. To administer the vaccine, the foil seal is removed from the applicator device. You can see the patch in the center of the device there in the middle image. It's then applied to the vaccination site, in this case on the upper arm overlying the deltoid muscle. The device is actuated by pressing the back of it. The administrator counts to 10 and then removes the product. That's the vaccination complete. So a very simple and intuitive process to receive a vaccination, but albeit quite different from what we typically do now with a needle and syringe. So I think I showed this cartoon back in 2021 that follows, and it's going to play through. Hopefully it's coming up on the videos. So the center of our technology is a patch. It's got this dense array of micro projections, about 300 microns across the surface, about 2,500 of them, onto which we dry down a vaccine solution. So when that micro projection penetrates through the stratum corneum into the epidermis and upper dermis, the vaccine is delivered from the projection into the skin, targeting the abundant immune cells present in the top layers of the skin. And why do this? I mean, I think we almost go a full circle back to 1796. Skin vaccination is a very, very effective way of generating an immune response. And I showed data and talked to some of these benefits that we see from our approach to vaccination. You can deliver more robust immune responses that has the potential for earlier onset of protective antibody responses, and also can use less vaccine to deliver the same protective effect compared to intramuscular vaccination. And that can have a significant impact on cost of goods for high-cost vaccines, but also for the number of doses that can be made from existing drug substance stock. It also, because we dry the vaccine down onto the micro projections, we remove the water from the formulation as we take a liquid and dry it onto our patch surface, we can improve the thermostability of liquid vaccines. And that in certain situations can enable us to take the vaccine completely out of the cold chain, either obviously at frozen temperature or 2 to 8 degrees more typically, and have that vaccine stored or distributed at higher temperature for a period of time, potentially for its shelf life, and other times for a shorter period to allow access and easier distribution, especially to those that most need it. Now, from the cartoon that I showed, or the series of photographs, we've designed this to be a very simple and easy to use device. It removes the needle, therefore improves the safety profile of vaccination. There's no risk of cross-infection, and there's no risk of needle stick injury to the administrator or the user. There's also the potential for lower trained healthcare workers to give vaccines, and also potentially self-administration as an option. And obviously with the intranasal vaccine now moving to guardian-based administration in the US, that sort of opens up the opportunity for vaccines to be given, especially for seasonal flu, in different situations outside the pharmacy, outside the medical clinic. And so it's good to see this as a predicate for new devices that can potentially also be applicable for self-administration. And also, I guess in the two areas, it has a potentially transformational benefit is in pandemic response. More vaccines from the same amount of drug substance provided faster in a range of different distribution channels, potentially the avoidance of people coming together to be vaccinated and exposing themselves to that disease threat. And it can make a huge difference in low and middle income countries, hard to reach areas where we can't currently get vaccines to those that most need them. So before going to the rest of the talk, I'll just take the liberty of looking back at what I presented back in 2021. This was obviously during COVID, when I spoke a little about the technology. On the call tonight, actually, a colleague of mine, Dr. Sandra Depolsinier, who did this work, looking at what happens in the skin when you have this dynamic application of a high density array. As those projections penetrate the skin surface, they disrupt the top layers of the skin, especially with the kinetic energy used to apply the device, and that raises a number of signals in the body, damage activated molecular pathways and potentially PAMPs as well, which when combined with delivery of the vaccine in a small area, we call co-localization, potentially contributes to the improved immune responses that we see. And we knew this back in 2021. It was actually very much the thesis for the technology back in 2011 when the company was formed. In that same talk, I also tried to look a little bit forward, was a little bit dangerous, and thought about the potential of our technology to combine with the mRNA vaccines, and what bringing those two things together, a new vaccine technology platform plus a new vaccination platform, could do for the reach and availability of mRNA vaccines. And I think that's something I'll talk a bit more about today. We've made some really nice progress on work with CEPI on a specific program around thermostabilization of mRNA vaccines. So it's good to be seeing that some of the things we were, I guess, thinking about in 2021, particularly during the COVID pandemic, have actually come to fruition in terms of programs that we're working on in Vaxxas. The other thing I mentioned during that presentation was the vaccine innovation prioritization strategy. This is back in 2020, and obviously a collective of hugely informed groups looking hard at how to improve the reach of vaccines for those that most need them. And this type of technologies and microarray patches or maps came out as the top of their list. Of course, this was back in 2020, and we're yet ready to see the technology space bring a product to market with a vaccine or progress beyond sort of phase 1, phase 1/2 studies. And I guess what I want to talk about a little bit today is what are those barriers. And I think back in 2021, it was generation of supportive clinical data to support the safety and the immunogenicity of a different way of delivering a vaccine through a patch, but also significant challenges with scale up of the technology from a manufacturing perspective. So I'll spend a fair bit of time talking around how at least we at Vaxxas are trying to address issues with scale of manufacture, but also costs of goods, which always runs in parallel to scale. So what I'll cover today, just that, by the way, there's a little bit of an introduction. I'll cover the sort of company overview, who we are, where we're based, a bit of our clinical data from our seasonal flu program, some data on the skin response that we see with vaccine patch administration, some information on ease of use, so trying to justify the claims around simplicity and potential for lower skilled healthcare workers or self-administration, some very preliminary market research data that we have. I do want to touch on our mRNA collaboration, particularly some areas of next steps and some of the stability data that we've generated, and then finally finish off with a run through manufacturing scale up. So a bit about our company, Vaxxas. We're founded in 2011 from technology coming out of the University of Queensland. We've been well funded through equity investment, but also non-dilutive funding through grants and partnerships. And we've got about 170 people, most of them based here in this facility in Brisbane, working on development of the technology in general. We don't develop new vaccines, we do in-license new vaccine candidates, but we work with either CDMOs or vaccine partners to take those vaccines through various studies toward the market. We've run a number of phase 1 studies in Australia. We've completed 5. We've got 1 ongoing at the moment. So we've essentially put about 524 participants through those completed studies. And we have a reasonably full pipeline with a bigger focus on manufacturing scale. A little bit on our pipeline, or the way to approach it. We have 4 sort of areas that we work within. We have our own sort of portfolio of assets that we in-license. So we have a COVID-19 asset in-licensed from the University of Texas at Austin and NIH, and also recently announced a RSV asset also in-licensed from NIH as well, and that was made public I think late last year. We also work with pandemic preparedness, so with BARDA, US government, on pre-pandemic influenza. It's our largest funded program, but also, as I've mentioned, with CEPI on mRNA thermostabilization. We partner with vaccine companies. We have a partnership with Merck, who's been established for a long time, and we also have partnered on seasonal flu as well to take that through to commercialization. Then I guess, the thing we can't forget about on the global health side, the Gates Foundation, World Health Organization particularly, have been supporters of Vaxxas for a long time. And we also now work with the Wellcome Trust on a polysaccharide conjugate vaccine in association with SK Biosciences. So we're just still working on those programs to bring our technology to those that probably most need it. So a bit of clinical data. This is data from 2018. It's reasonably old, but it was our first clinical study which showed that lower doses with patch application can generate similar immune responses to a standard dose given by needle and syringe. So this compared a single monovalent flu antigen, H1N1, delivered by the patch, compared to the same antigen in the commercial trivalent vaccine at the time. So on the left-hand side, when we compared similar doses to IM given by the patch, in this case applied to the upper arm or the forearm, so UA or FA, we saw from the HAI readout a sort of significantly higher titers at day 7, day 21, and day 60 compared to needle and syringe shown in the grey. We obviously had a placebo component as well, which is a placebo patch shown in the turquoise. So that was similar doses. On the right-hand side, we used a 1/6 dose, so 2.5 micrograms compared to 15. And you can see the green trace there, essentially overlying the responses with a significantly higher dose given by needle and syringe. Now, it was really encouraging. It was the first clinical study with a patch technology to show that lower doses could generate similar responses to full dose given by intramuscular injection. Now, since then, we've partnered and we've done a study with quadrivalent influenza vaccine. All doses delivered a single patch administration to the skin. This was 150 subjects, phase 1 study run in Australia. And in these plots, I'm showing the 4 different strains of that quadrivalent influenza vaccine, the 2As and the 2Bs, and showing just one of the doses that was looked at in the study with the patch. So in this case, the lowest dose, which was 1/3 dose of the standard intramuscular dose. So 5 micrograms of each strain within the combined product. And again, across all strains, you can see the turquoise trace showing the responses with the patch compared to the commercial product containing exactly the same antigen shown in the grey, and then a placebo group shown in the red. So very, very similar responses that we saw with our monovalent study, but in this case, even at 1/3 dose, we were seeing statistically higher titers than we saw with a higher dose by intramuscular injection. And I thought I'd just compare that to some data recently from Moderna. This is from their phase 3 study. It's published data showing the comparison of their titer, sort of the geometric mean titers between their vaccine and the comparator in the study, which I think was Fluorix, across the 4 strains in the study. So looking at the sort of essentially the fold increase that you're seeing across those 4 different strains with mRNA vaccine. Obviously, it's a larger study run on both adults and older adults, but when I compare that to the titers that we saw, or the GMTs we saw in our study across the various strains, we also saw significantly higher sort of GMT ratios at all doses with all strains with our technology at day 21. So it may show the potential for similar or reduced doses to give a sort of more significant immune response than the standard of care standard dose flu. It could also open up the opportunity in older adults as well. But I must say, all our data at the moment is in the adult population. No work so far in older adults, which we're looking to move to. So just thought I'd share that flu data with you from a recent non-published study, but also the skin response as well. So this is not actually from that flu study. This is from a COVID-19 recombinant protein phase 1 study with a spike protein antigen. And the reason I'm showing this data is because in this study, we actually applied 3 patches to each individual. Some of those patches contained vaccine and some were uncoated, so placebo patches. In this particular individual, they received 1 active patch and then 2 inactive patches. So on day 3, you can clearly see the response from the active patch compared to a patch with nothing on it. Maybe if I just get my pointer here, the active patch and then the 2 placebo patches. By day 7, all those sites are starting to resolve, but the active site remains more apparent. In this case, for this subject, by day 90, all application sites had resolved. So we do get a local skin response, not unexpected. We're putting vaccine just below the skin surface and getting a cellular response to that vaccine being delivered. That skin response typically completely resolves by about 2 months post-administration, but can persist for slightly longer in certain cases, so up to 3 months, and it's typically either a slight hyper or slight hypopigmentation. I must stress that this is typically barely visually perceptible to the investigator and subject in the study. Just a comment, all our phase 1 studies so far, apart from one we're currently running, have looked at non-naives, so measles, rubella, flu, COVID-19, and also show some market research data we've put together to look at people's response to that skin response, if you like, in terms of their views on skin-based vaccination with our technology. But just a comment, we do need to look at larger numbers to fully map that out. So I showed this slide before. It's the same hypothesis as to why we see a skin response. I just sort of show a reasonably high magnification image of a patch application from that COVID study, and you can see the individual sort of puncture points in the skin at day 3 through camera. So you're getting very, very localized redness, which looks like a erythema, broader erythema when you go to a higher, a sort of a lower field of vision. So it is almost individual erythema responses associated with that localization of antigen delivery. I'll probably skip this next slide a little bit. This is actually work that Sandra, who's on the call, did from our flu study. We took biopsies in our first in 2018 from individuals. We took biopsies at day 1 outside the patch application area in both subjects that received an active monovalent flu patch and a placebo patch. And you can see the H&E results showing infiltration, especially with the active patch at day 4. And that resulted in cellular infiltration to that area and immune cells, which you looked at through flow cytometry of those biopsies at those different time points. So we're actually getting an active response. We've continued to look at skin immunology. We have a collaboration with the University of Queensland through Associate Professor David Muller and also one of his postdocs, Dr. Chris McMillan. We're starting to use some more sort of sophisticated tools to look at what's happening in the skin, both of animals, so mainly in mice, but also some limited clinical work without vaccines. And by using sort of spatial transcriptomics with different degrees of resolution, they can get a real idea of what's actually happening in the skin in response to patch administration of a vaccine and what sort of pathways are being triggered and how does this compare to, say, intradermal delivery as well. So some interesting research that should give us more information about skin delivery of vaccines. So shifting gears a little bit, I wanted to touch on work with ease of use, human factors. So I'll show a video in a minute. This is a video from a clinical study conducted in Australia in adults over the age of 50. I've changed that from older adults because I don't feel that I'm old, and up to the age of 90. And in this study, there was no vaccine contained in the combination of product, but there was a patch applied during this clinical trial. And we asked participants to follow a simple instruction for use document, and it's shown on the desk in front of the participant, and apply the product to their upper arm following those instructions. If I just play the video, so this participant consented to this video being shown. She's removing the foil seal. It's an early prototype, so we're just working on that aspect of that foil seal a little bit to make it easy to remove, but still have container closure integrity. She's applied it to her upper arm. She's actuated the device. She's now going to she's counting to 10, and then she's going to remove the product. This participant did a fantastic job of following the IFU. Not all participants did such a good job, and so we're gaining that data, I guess, from human factors and some of these formative human factors studies about ease of use of the device. We've conducted these trials also with vaccinators, pharmacists, doctors, nurses, to make sure that we can learn about sort of the interaction of the device through experienced and then inexperienced users of the technology. In terms of market research, this is data from some market research done in the US by Incline Solutions Group, who were contracted by Vaxxas to undertake this work. And this was looking at the seasonal flu context around whether people would, they were shown images of our product, they were shown images of the skin response from our studies, and they were asked a series of questions about their views on this new technology in terms of vaccination uptake. And we divide it into two populations, sort of younger adults, 20 to 59, and then adults 60 years and over. And the first question was if you could choose between our technology and a shot, which would you choose for your vaccination? You sort of, you definitely choose the needle-free, probably choose a shot, definitely choose a shot. And overwhelmingly, I think, I can't quite see it on the screen, about 87% of young adults said they'd probably or definitely choose a patch, and about, how many was it? About 77% of older adults said they'd probably or definitely choose a shot. I choose a patch rather than an intramuscular vaccination. And then the next question we asked was actually if there was would you actively seek out a location offering this technology rather than receiving a standard vaccine through needle and syringe? And again, overwhelmingly, most people said yes, they would actively seek out a location offering this technology for vaccination for seasonal flu. Again, this is US data from a broad range of demographics. So that's very encouraging in terms of the potential of this technology to maybe overcome some of these challenges we're seeing with seasonal flu uptake in the US and in high-income countries around the world. Moving on now to our mRNA program, and this was actually, again, work that we sort of first started in association with the University of Queensland through a specific grant, looking at the fit between mRNA and lipid nanoparticle vaccines and our patch technology. So we had some initial data, we then had discussions with CEPI, who funded a program with us to look at the ability of our technology through that drying approach to stabilize mRNA vaccines. And most of this work has been done with mRNA encapsulated in lipid nanoparticle formulations. So we completed our stage 1. That was in collaboration with a few groups in Australia, the base group at UQ, Griffith University, Professor Nigel McMillan, and also the Muller Lab. And now we're collaborating as well with SK Biosciences on Japanese encephalitis virus vaccine development with mRNA. At the start of the year, we were awarded a prize from BARDA as a Patch Forward prize, specifically looking at mRNA technologies, and we directed that towards our Hexapro mRNA vaccine candidate, and we're continuing to work on that at the moment with the UQ base to push that through. So some data from that program, and again, Sandra, who's on the call, has been heavily involved in generating this data from our sort of preclinical early stage team. And what we're seeing here is a patch on the left-hand side coated with about 25 micrograms of mRNA contained in lipid nanoparticle at a ratio of about 1:16 or around that in terms of mass loading. So that you can see the micro projections there. Again, I'll just grab my pointer. You can see the coating on top of the micro projection and a reasonably good and uniform coating of that material dried onto the micro projection. So each one of those is about 300 microns in height from tip to base. On the right-hand side is some of the stability data that we've generated using mRNA in both Moderna-like, and this is Moderna-like data, and also Pfizer-like lipid nanoparticles. And this is measuring the change in relative encapsulation efficiency following drying of the material onto the patch at different temperatures. So it's nice and obvious. So the coldest is blue, 2 to 8 degrees. Up to 25 degrees, 60% RH in the green, and then in the red, we're seeing 40-75 data. And what we're seeing at 2 to 8 and 25 in particular is great maintenance of the encapsulation efficiency. We do run a lot of other methods alongside this, looking at particle size, looking at integrity of the mRNA within the lipid nanoparticle, and also in vitro expression of that material in cell culture, of the protein in cell culture. So 2 to 8 degrees and 25 degrees, with the formulation of that material on our patch, we're seeing a really, really positive stability. I mean, even at 40 degrees, and I think we've actually got some better data than this now, we're still seeing reasonably good stability performance out to 60 days. So that's 2 months. So that could really open up the opportunity to get mRNA vaccines to those areas where cold chain maintenance and distribution is very, very challenging. So really pleased with this initial data and hoping to now move that into our JV program and generate similar data, and also generate some immunogenicity as part of our stability program as well, which I guess would be the real sort of unequivocal demonstration that you've got good stability performance at a range of different temperatures above -20. And I think there's a good potential in Australia. I mean, it's kind of, there's a real sort of emphasis on mRNA vaccines in Australia. There's some great academics working in the area. We've also got large companies looking at Australia, setting up operations here as well. So our next steps, around our sort of program with CEPI to generate preclinical data by immunogenicity with the JV construct, and also with Hexapro. We're working on our analytical capabilities to make sure we can also characterize the lipid nanoparticle chemistry a little bit more. This could be important for stability. With UQ, we're also investigating where protein expression occurs in the body, in preclinical models in the mouse, and also what type of localized cell responses we see, again, through some of that spatial transcriptomics work that I referred to. And that could be really interesting to see what we see in terms of where we get expression. We've done most of our work with Moderna-like or Pfizer-like LNPs. We are interested in different lipid nanoparticles, which may be better suited for skin delivery with existing and new partners. And looking forward to move into clinical trial production, especially in our CEPI program with our partners here. And obviously, as we generate this data, looking to showcase that with a range of different vaccine companies who work in that space. So just shifting gears a little bit now, I'm going to make sure I don't run out of time, just to our manufacturing capability. This is our facility in Brisbane. For those that know Brisbane, there's a city, there's a river, there's a facility. It's about 5,500 square metres. It's our R&D home, and it's also going to be the site of our first commercial production. And the building was funded by Queensland State Government. We are a tenant in the building. I'll just skip through that. So what do we have in the building? We have laboratories, we have clean rooms, we have warehouses, we have offices. To enable us to keep on manufacturing product for phase 1 studies, but also to move into phase 2 and phase 3 manufacture, and then later commercial supply. So we have all the necessary sort of tools and capabilities around the device component, the applicator, the patch, and some of the other plastic parts that make that up, but also the ability to make the combination product under aseptic manufacturing conditions. And this facility will undergo a TGA audit to make sure we're compliant with CGMP for phase 2 and phase 3 supply later this year. So our manufacturing is really in 2 parts. So there's the device manufacturing, so making the patch, the applicator, bringing those things together, having those device components sterilized through gamma irradiation, then bringing in those device components into our aseptic manufacturing process, where we combine the vaccine formulation onto the patch and have the finished good as the end product. I'll just skip through the device stuff quite quickly in the interest of time. This is a sort of a blow-up of our device. It's pretty simple. There's a shell made from aluminium, made by a company that make coffee pods for Nespresso at massive volume and low cost. There's a spring, which is a silver part, which we make in-house, which provides energy for the patch to engage with the skin, which projects to penetrate through. There's a desiccant component that keeps the interior of the entire device dry. As a core piece, there's the patch, which is obviously micro-molded, and then there's a foil seal, which is subject to container closure integrity testing through helium leak testing. And it's intended that this device is a primary pack, and there's no need for any secondary overwrap to maintain the integrity of the product. Look, I'll just show how it's made. So the patch itself is made by injection molding. And we've moved up now to 4-cavity molding. Make a little bit of noise for the video. Turn that noise down. So you can see at the moment we've moved up to what we call 4-cavity molding. So each shot from the molding process makes 4 parts, and we're just automating the molding process at the moment. This is just our initial 4-cavity molding system, and the camera person is going to go around the other side. They're going to open and show you the molding process. It's basically injection of plastic at liquid plastic at high speed into a mold. So the mold's going to close. You have tools going to close. Plastic's being injected. The tool's going to open, and the part's going to be presented as the 4 patches. So just gives you an idea of speed of production. And then there's a robot that picks out the 4 patches and then pulls them apart from the runner and the sprue to produce the individual device components. So very, very suited to high volume manufacturing. The applicator itself has to be assembled together. We do that at our facility here in Brisbane. It has a number of different parts which come together to make the finished applicator without the patch, which goes into trays, which are then subject to sterilization through gamma. It's a very simple process. We have a manual process at the moment in a clean room, which will be grade D, and a series of equipment that enable that. But we are in the process of automating that manual assembly process with a company called Rickiger in Switzerland to bring those steps together and to produce about 60 sub-assemblies as an input into aseptic manufacturing, and that machine is being built at the moment. The next slide just shows you some pictures from Switzerland of the machine under construction. It's a magnetic conveyancing system. It's non-linear, so each of these sort of robots can move at different stages throughout the process, moving goods from part to part. But this enables us to basically have a fully automated device assembly system, which will come to our facility at the end of this year. Right, since it's the vaccine audience, I'll jump on to the aseptic coating process. So again, this is done under grade A conditions and subject to aseptic process qualification. So sterile vaccine formulation, sterile incoming device components. We're scaling that up, and scale is exceptionally important for cost of goods. So on the left-hand side, you can see cost of goods of the device, excluding vaccine costs, but including device components and the aseptic manufacturing component. And we're trying to compete here with a pre-filled needle and syringe, which you only do through significant volumes at about 20 million units per annum. You get down to about a similar cost point to a pre-filled needle and syringe. At the moment, we have a manual aseptic process. I'll show you a video of that, but we're moving to semi-automation at the moment, and we have a line that's completed factory acceptance testing in Switzerland and is ready to be sent to our facility here in Brisbane. So it should be installed and qualified this year, and that'll increase our batch output from about 500 finished product units per day, which our manual process does, to up to 10,000 parts, finished products per day from a single batch with semi-automation. And obviously, to get to the full scales that we need to fully automate the process. The obviously other significant advantage for an aseptic process is getting operators out of the manufacturing area, because even though we have very, very well trained operators, they are the most likely source of product contamination. So removing them from the manufacturing space is our plan. So just before I show you the video, I just talk a little bit about how we put the vaccine onto the patch. So it's done using a printing system. There's a print head, there are patches, there's a camera. So the camera images the patch, checks the location of the patch relative to the print head. The print head then aligns to each patch and prints multiple drops onto the micro projection. So obviously we dispense about 400 to 500 drops per dispense, covering a fraction of the array, and then we do the same thing, and we do this very, very quickly with very high accuracy and precision. So you can see drops from the print head. They're about 150 picolitres in volume, and then they are basically drying rapidly onto the micro needle surface. There's no treatment of the microneedle surface. It is a plastic. It's reasonably hydrophobic, so the drop sort of sits on the tip of the micro projection. Obviously, if it drops down towards the base, it won't go into the skin, so we need the material on the top half of those micro projections. This is technology developed in-house by our very, very talented engineering team. So this is the manual process in the clean room. So I have this video. I don't know if it's going to work very well on the webinar. This is sped up a little bit. This is the camera in our clean room. It's a grade B clean room. It's been qualified at the moment. This is a mock training run for our operators who are gowned up for grade B, grade A working. We have a grade A zone, which is underneath the laminar downflow cabinet with some degree of restricted access through perspex shielding to prevent the operator from ingressing over the critical zone. So on the left-hand side, we have the printing approach. So you can see the printer working very fast here, sped up with trays of patches. Those patches are then inserted into the applicator, and then the final step is to seal that with a foil sealer. So this is a foil sealing machine, places the foil lid on top and seals that to make it integral. It's actually, in this case, because we need to qualify it for media fill, there's also a media fill dispensing station, which is not typically involved in the process, but adds media to each vial so we can qualify the process aseptically. So what we've got with us, this has 3 operators working at the moment, a very skilled operations team. We were removing 2 of those operators from our next line, which is semi-automated. So if I just jump to the next slide, it's a little bit corporate, but it should play. So this is a video from Switzerland, from Rickiger, who are doing the automation of those 2 steps. So at the moment, the machine is running at low speed. It'll speed up. The trays of coated patches coming in, the robot will pick those up, insert them into the applicator. Then there's a QC step with a vision system to make sure there's no defects and the patch is located properly. That'll show up in a minute. You'll see the robot arm start to speed up now to normal operation speed. And this produces about 12 finished parts per minute and potentially up to 10,000 units per day. This is the camera system inspecting each patch to make sure it's located. It's a foil sealer. And obviously we've removed 2 operators. So we're really, really excited about this technology. It's on the way to our facility here, and will be significant for our phase 2, phase 3 manufacturing campaigns. I should just say it's contained within a restricted access barrier system. It's a system being built by Scan. It's currently undergoing factory acceptance testing in Japan, and will also be shipped along with the semi-automated line to our facility. So last couple of slides. This highlights things we're looking forward to in the year to come. Obviously, having the facility accredited by the TGA for investigational product manufacture under aseptic conditions. To continue our work with the US government on pre-pandemic influenza, and we're looking to move now to phase 2 studies once we have that TGA license, and in particular with our lead candidate, seasonal flu, and aiming to have a product to market and license by 2028. So we've got a bit to do before we get to that point, but all the signs are pointing very positively. And that's my last sort of slide for Vaxxas. I did want to make, since I've got a few people online, a plug for if you want to know more about microneedles, the world's largest, most comprehensive meeting on microneedle technology is coming to Brisbane this year, May 11th to 14th. We are co-partners with the University of Queensland, and it covers all aspects of microneedle technology for human health, including diagnostics, small molecule delivery, biologics, vaccines, but also some cosmetic applications as well. So yeah, if you are interested, Microneedles 2025, and there's still opportunities to join the meeting. And so with that, Branka, I think I'll finish the presentation, and happy to take any questions that have come up during the presentation.
Thank you, Angus. That's absolutely fantastic. And it's been amazing to actually see Vaxxas in the last decade grow from strength to strength. And, it really is, one of the leading biotechs, or the leading biotech here for us in Australia. So we take a lot of pride in your success as well. So
thank you.
Thank you for that excellent presentation. I'd like to invite you all to please post your questions in the Q&A, and maybe I will start with big advances and big clinical trials and phase 2 clinical trials that you are planning, and your manufacturing and GMP in the new facility. What do you think will be your first product that will come to market?
At the moment, seasonal flu is our lead candidate, looking to go into phase 2 studies at the beginning of next year in both adults and older adults. And then, following behind that, we're working with the Gates Foundation on measles, rubella, and looking to move into a phase 1/2 study in the Gambia. There's already been some excellent work done by another microneedle group with measles and rubella in the Gambia, and we've done a phase 1 study here in Australia as well in healthy adults. So 2 lead candidates. Also looking to move forward with our RSV asset as well, and particularly into older adults. So that has not yet been into the clinic, but should go into a phase 1 study next year. And I think where the vaccine is either generally known to be safe and efficacious as an injectable, it really does help accelerate that sort of timeline to market and decrease the risk of that development as well, which is why we're sort of picking those sort of known vaccines as first products.
Oh, fantastic. Would it be fair to say that you would probably go over the recombinant protein combination first, and then as you work on the stability of the mRNA and so forth, advance that later on?
Yeah, so mRNA, and it's interesting, we've got some really nice thermostability already. It's really encouraging. We're still have done some lovely preclinical immunogenicity with mRNA. Whether we'll see the benefit of dose sparing with mRNA in the skin, I think is still an open question. We've had some quite nice early data, but preliminary studies, and look forward to continuing to develop that. But I think the real advantage there is potentially this thermostabilization benefit, but also the, if there's any sort of benefit from delivering to skin from a, expression of protein. But I think it is more complicated, as you mentioned, and that will be a new vaccine, depending on what target we go forward with, then it could be a more complicated clinical program, and obviously a new vaccine carries more technical risk from the vaccine side as well as the delivery side as well. So you're absolutely right. Sort of starting with, a protein-based vaccine is a really good place to start, especially one like seasonal flu, which is well understood. You can measure a very specific immune response in your early studies, and you're going to hopefully move towards a decent protective response. And that's why we've focused a lot on seasonal flu. Pre-pandemic influenza is interesting for us. I mean, that is, an area where we can, again, pandemic response, and flu pandemic vaccines are stockpiled, especially for flu around the world. And the challenge there, of course, was the immunogenicity in a naive population. So we have a large phase 1 underway at the moment in Australia, about 258 subjects, looking at both unadjuvanted and adjuvanted patch administration to those participants compared to the standard of care, which is adjuvanted IM. So it'll be interesting to see what our sort of day 43 readout is post-dose 2, and whether we can get anywhere close with an unadjuvanted technology in what has typically been a space where you need an adjuvant to elicit a protective immune response for pre-pandemic.
Fantastic. So I'll go through some questions that we've got. So Rafa said, thank you for a great talk. Did you measure the mRNA stability after storage on the applicator? You said you measured encapsulation efficiency, but I'm not sure if that is a measure of the stability.
Yeah, good question. So yes, it was within an applicator device. So what we do is we coat the patch, we dry it down, we put it into our applicator device, we seal it and we store it. And look, we have done some other measures of stability. I thought I'd show the encapsulation efficiency. We also look at mRNA integrity, the particle size, and you see at 40 degrees, the particle size distribution start to significantly change as that encapsulation efficiency comes down. How particle size actually then relates to actual effective expression of the mRNA when delivered by skin, I think that's still a little bit uncertain. But we're also running in vitro expression as well. So it's another assay. So when we extract the material off the patch, so we dissolve off the coating that's on the patch into liquid and put that into a cell bed, do we get expression of that protein? But as I think I mentioned, the best test would be to run some preclinical immunogenicity at different time points and show no difference over time in that immune response, within the caveats of the broadness of the immune response that you'd probably be measuring. But I think encouraging early data. And I think the great thing about mRNA LNP is once you've shown it, hopefully for one LNP with a certain payload of mRNA, albeit maybe variants in the sequence and size of that mRNA, that should be applicable across all of those different candidates, right? So that's the real advantage. So that's why it's exciting to be doing it now.
Okay. Another question that we have is, with the skin intradermal delivery, have you looked at mucosal immune responses?
We have, both preclinically and clinically. I think clinically, in a study in 2018 with a Singapore monovalent flu, we looked for IgA responses in saliva. Didn't see very much in a small number of subjects. We did the same thing in a COVID-19 study. Again, from the clinical samples, we didn't see a lot of strong indication of a mucosal response in the samples that were collected. Preclinically, we have, but I think we're also now focused a lot on what is the differential immune response in the skin that you get with patch administration, say, versus intradermal delivery by needle and syringe as well. I guess the hypothesis is that skin is being a mucosal surface, it might translate into lungs and stuff like that from a protective response. Clinically, I think we need larger studies to actually look at, do we see any sort of mucosal response in saliva, say for flu, nasal swabs, whatever the target would be. It's something we're aware of. We have looked to see it, but we don't have strong data to indicate we're seeing that at the moment.
Yeah, thank you. Can you speculate on how the patches will work in a naive population? And what are the Vaxxas plans for other pediatric vaccines? What considerations affect these decisions?
So naive population, I guess our pre-pandemic influenza study will be the first study clinically in a naive population. So we haven't had a readout from that yet. It's a 2-dose study, prime and boost. There was nice work done by Micron in the Gambia in infants with measles and rubella. They got sort of very similar immune responses with a single dose in that population to subcutaneous injection of a commercial comparator. So I think I don't see why it wouldn't be effective in a pediatric population. I think one of the problems in pediatric vaccines is the number of combination vaccines that you have, and obviously, to develop a combination vaccine like a pentavalent is a lot more work than doing a single disease. Even flu is 4 entities, right? But so that's kind of is the challenge in pediatric vaccines. Often is kind of to make that change from a needle and syringe to move to a patch version of a multivalent vaccine is a significant undertaking from a clinical perspective, and also even from a CMC perspective. So something that we're not pushing hard at the moment, but certainly for measles and rubella, that target there is the infant population in low and middle income countries. Similarly for typhoid as well, because that's where it can make a difference. And with our typhoid program, again, it will start in adults phase 1 and hopefully move into infants in country. I guess I didn't completely. I think obviously that removing the needle. I mean, teenagers are an interesting market as well. Like HPV vaccination is a potentially really interesting one. From a teenage demographic to encourage vaccination rates with a non-needle based technology, especially in Australia when we do school-based immunisation. We all know of stories of teenagers not having their vaccine because of sort of mass hysteria and contagion down the line. So that's an area we are really interested in, as well as the potential, maternal vaccination by midwives could be a really good option too. So midwives can give vaccines, especially in New Zealand, I think, and I'm not sure Australia, but they don't typically because of some of the challenges with need to respond quickly. But that could really make it much more straightforward for a midwife to give a maternal vaccination.
Yeah. One of the questions that we do have is, can you speculate on the possible reactogenicity in humans of an already reactogenic mRNA in the skin?
Yeah, good question. I mean,
all the patches that rely on creating skin damage.
Yeah, I mean, Moderna, BioNTech and others have looked at intradermal delivery of mRNA lipid nanoparticles. You will get some skin response associated with that lipid nanoparticle component in the skin. We've only done preclinical work to date. We've seen degree of reactogenicity with standard vaccines in the skin, as you would expect to see. Whether that's going to be exacerbated by lipid nanoparticles, I guess we need to find out. I mean, pre-pandemic influenza program does include an adjuvant, a very, very low dose adjuvant, and it does increase the skin response, but not to a point where it's of any sort of visual concern. So, I mean, I think we need to run the studies, and that's also why I guess we are looking at maybe other alternative LNPs that may mitigate some of that effect. But that's a big development project to start looking at new LNPs as well as a new delivery way system for existing formulations. So, yep, I mean, that's, I think that's, I mean, I guess it's whether the benefits in terms of thermostability, ease of use, rollout outweigh the sort of the skin response that's seen. Yep.
Yep. And another question is, do you use GMP grade mRNA LNPs?
So at the moment, it's a big question I'd probably need to come back to. Most of our material that we've had made has been synthesized by partners, especially UQ base, to a high standard, but it's not made under GMP conditions. Obviously, as we move ahead with our CEPI-funded program with SK Biosciences, they are going that we will receive GMP quality material for that program. And as I mentioned, I think understanding the lipid chemistry and control of that lipid chemistry and assessing not just the mRNA quality, but also the lipid nanoparticle quality as alone, aside from the combination of the two, that's something we're looking to expand our degree of sort of analytical capability in, and also working with our partners to try and to do that to make sure we fully characterize the materials we put into preclinical studies. Yep.
Yeah. And the patches themselves, do they have any biodegradable or biocompatible stabilizers for the mRNA in the vaccines?
So the patch itself is made out of a biocompatible polymer. It's a high melt flow polymer, and we put the entire product through biocompatibility testing. So there's no treatment of the patch post molding, apart from there's a sort of we'll call it blow down step to remove any particulate during the grade D molding of the patch. There's no surface treatment. We have looked at surface treatment to make it either more or less hydrophilic or hydrophobic. Yeah, that's absolutely possible to do. That's very simple ways of doing that. But at the moment, it's a reasonably uniform plastic surface that we coat onto with no modification for the vaccines that we dispense. So look, same substrate for lipid nanoparticles as it is for a protein-based subunit vaccine.
And
But obviously, we formulate with excipients to overcome challenges of dispensing and drying rate, for example.
yeah, well, while you're mentioning the drying process, one of the questions is actually during the coating process, does the drying affect or damage the vaccines, and how do you prevent that?
Yeah, that's exactly right. So we have the way in which we coat with this small drop deposition approach. It's very, very fast evaporative drying. So it's an evaporative drying process. There's no volatile solvents used. It's aqueous. There's no exaggeration of that drying through heat or changes in pressure. So it's a purely an evaporative drying process. But our drop volume is about 150 picolitres per dispense. So even as you're imaging over 70 microns, you can see the drop drying because it's such a small volume. So it's a combination of that drying approach plus the use of certain stabilizers, mainly things you'd use in lyophilization, that contribute to that dry down stability. So we look both at the immediate dry down effect, but then obviously the stability as well, because you could get a hit on dry down but have a very stable product. But what we try and ensure is that the dry down stability is as high as possible, so that we're not wasting vaccine during that drying process. And certainly for most vaccines we've worked with, even with mRNA lipid nanoparticles, we've had very good maintenance of encapsulation post-drying as well, so 90-95%, and then maintained it on stability. But that can vary a little bit. Live viruses, measles and rubella, you do lose a little bit of live virus as you dry. Yep.
Yeah. And then we did have a question about the self-amplifying RNA. Do you have any plans to integrate them with the patches?
Yeah, we've definitely thought about it. We haven't done anything in that space yet. It's a really one of the challenges with mRNA lipid nanoparticles is the amount of LNP you need to deliver the dose of mRNA. So, 25 micrograms on a patch may not seem like much, but actually you've got a lot of LNP there to dispense that. So obviously with self-amplifying, you can potentially reduce that ratio, which should be beneficial from a manufacturing perspective. But we have not done any work in that area yet. But we obviously would. We've got a lot of things on our plate, and that's the other thing, kind of the focus. You can do. There's a huge number of interesting experiments you can run, but trying to get things, push things towards the market, you have a good focus. But we have good relationships with UQ in particular and other groups who can maybe do some of this work for us as well.
Yeah. And some of the final questions is, what are the considerations for populations of color? Would the color and the skin composition have an impact on vaccine taken efficacy?
Yeah, so great. I mean, I think a couple of points there. I guess one is aging of skin. So there's also not just the sort of the skin tone, but there's also aging of skin from early infant skin right through to older adult skin. And we know about immunosenescence in the older adult population, how that affects the dose you need in the skin. We haven't done an older adult immunogenicity study, but we'll do that phase 2 with flu. From a skin tone perspective, we have collected Fitzpatrick skin tone data from our current studies. Obviously, those have been done in Australia, so they tend to skew towards a Caucasian demographic. But we've also done some work in darker skin tones to look at the nature of that response that you get and whether you're more inclined to hypo or hyperpigmentation. Look, so far, there doesn't seem to be sort of increasing propensity, but again, we've only done phase 1 studies, small subject numbers. MR would be in the Gambia, so it would be in high sky skin tones. So that would be, the first. And we know we've seen data from Micron in that space as well, with no difference from what they saw in their sort of US-based studies compared to what we saw in the Gambia. So that's encouraging, I think. But that skin response will still be sort of visible for a short period of time.
Yeah, thank you. And so I think our members have asked all the questions, but before I thank you, I would just like to ask you if actually people are interested and if there are opportunities or capacities, how they can best contact you for collaborations or any interest.
Yeah, I'm on LinkedIn, and I know you think you've got my email address. You can also make an inquiry through the website. We have actually do look at our inquiries through the website. So if you do make an inquiry through the website, one of our BD team will pick that up, or our comms team. But yeah, I'm on LinkedIn. That's easy. Or it's a Forster@vaxxas.com is my email address.
Yeah, wonderful. Thank you so much. Thoroughly enjoyed your presentation. Good luck with all the projects. We look forward to continuing to listen to Vaxxas' success. And thank you once again.
Yeah, thanks, Branka. A real pleasure. Thanks for joining.
Thank you, everybody, for joining. Till next time.
Bye then.